Good morning, good afternoon, good evening, everyone. We're going to start in just a few seconds. We're going to give people a few more minutes to join, but we are so happy that you are here with us today. All right, we are right at 1 minute after noon on this beautiful day here in Chicago. My name is Alyse Mauro Mason, and I am a volunteer event director with Swim Across America in Chicago. And I also have the honor of being your moderator today. I am joined here by 6 doctors and researchers from Rush University Medical Center that have all received grants from Swim Across America Chicago. We are so grateful that they were excited and willing to join us in this cancer conversation with all of you today. Our discussion will last about 45 minutes, and we will leave 15 minutes at the end to take questions from the audience. So please, at any time during this discussion, use the Q&A option in WebEx to submit your questions and at any time, and then we will address them in that last 15 minutes. If you would like a specific doctor to address your question, please also include that in your question when you submit it. It is now my pleasure to introduce you to our panelists from Rush that are here with us today. Doctors, thank you for being here to share your passion about cancer research and what the Swim Across America partnership means to all of you. As a reminder, all lines will be muted when you are not speaking, so please remember to take yourself off of mute before you begin speaking. Panelists, we will go in alphabetical order for introductions, so please share the following with our audience today: your name, your title, your role at Rush, the nature of your cancer research, and how Swim Across America has made an impact on your work. So we are going to start with Dr. Barua today. If you could please come off of mute and share those 3 things with us.
Good afternoon, everyone, and thank you very much for joining. I am Animesh Barua, an associate professor at the Department of Anatomy and Cell Biology. One of my research area is ovarian cancer. As you may know, this is a very fatal and aggressive cancer of women, and it is associated with very high rate of death. And because there is no effective early detection test, patients with ovarian cancer in most cases are detected at later stages, during which the rate of mortality belongs to 90%. However, if we can detect this disease at early stage, we can save most of the patients and their lives. So our work, one of our main work is to establish an early detection test. So currently we are working to find a panel of blood markers, and we are developing an ultrasound imaging so that using both blood marker and ultrasound imaging, we can detect a woman at early stage of the disease. So with our swim across funding, we have able to establish a panel of markers. Now we are examining how this marker correlates with ultrasound imaging so that if a woman have this marker in her blood, we can ask to go for ultrasound monitoring so that we can catch the disease when it is remaining in the ovary. And we go for our treatment modality or our surgery. So in doing so, we are using an animal model that resembles very closely, remarkably, to our human patient. And the data so far we gathered from this swim across funding allowed us to apply for federal funding like NIH, Department of Defense. And by my previous grant, I have successfully funded from NIH and NCI, National Cancer Institute. So I am really very much indebted and grateful to the funding of swim across. In the long run, it will allow us to devise some detection and treatment modalities so that we can save women in this country as well as globally. And so far, we have published one paper from this swim across funding. So I will be here to answer any question. Please feel free later when you have any question. Thank you very much. Once again, we are grateful to swim across for their funding. Thank you.
Thank you so much, Dr. Barua. We're going to pass over the mic to Dr. Bishehsari.
Hi, everyone. My name is Faraz Bishehsari. I'm a gastroenterologist, Associate Director of the Rush Center for Integrated Microbiome and Chronobiology Research. I'm an Associate Professor in Internal Medicine. My studies are focused on mechanisms involved in gastrointestinal cancers, focusing on colon and pancreatic cancer. Particularly, I'm interested in how lifestyle and risk factors in the environment promote cancer via interacting with the environment around the cells. Cancer cells in the tissue, so-called tissue microenvironment. To study such complex topics, many labs, including mine, has been using animal models, but in order to make it closer to a personalized approach to the bedside, I'm particularly interested in moving the field forward using primary cells from human tissue. And in this regard, our lab has been developing and using organate models, which is patient-based models from mouse, and also from patients' human tissue. And part of those studies had the support of Swim America. And we published several papers with the support of Swim America. And we are applying to NIH. My current NIH funding also was based on some data that I had produced with the help of Swim America Foundation. Thank you.
Thank you so much. And now, because Dr. Barua and yourself both just shared that early stage funding from Swim Across America enabled you to get that NIH funding, would you mind just sharing a little bit more about why early stage funding is so important in order to advance that research?
Yes, because the reason is if we have that funding, we can transfer our information that coming from this swim across funding to a larger clinical study using a large cohort of patient samples or patients, so that we can, in our next step, with the information and the ultrasound imaging agent we are developing, we can apply to our clinical studies, and it will give us a early detection test, or we can say a screening test, so that we can identify a woman even before the tumor becomes solid mass in the ovary, so that we will have a subset of women who are at risk with developing this cancer, and it is greatly helpful. It will for saving the life of women. So we need funding for our clinical phase studies. That's the reason. And with the use of this basic information that got from Swim Across, it will help us for a larger funding from federal grant sponsor like NIH and NCI and Department of Defense. Thank you.
Thank you so much. And for our audience, what does NIH stand for?
National Institute of Health. It is one of the biggest and our public sector funding for all of us. Thank you. It has many institutes.
Thank you so much. We are going to pass over the mic to Dr. Blank.
Hi there, thank you. Thanks everyone for joining, and thanks to Swim Across America. My name is Alan Blank. I'm an orthopedic oncologist here at Rush. Been here 5 years. I have a musculoskeletal oncology practice. So predominantly I take care of sarcomas, which is a type of cancer in the bone, cartilage, muscle. But also I have an interest and a passion in treating patients with metastatic bone disease, which is cancer from other organs that have spread to the bone. I was fortunate enough to be part of a grant led by Dr. Pratap, which you'll hear from shortly, evaluating how these metastatic cancer cells, in particular breast cancer cells, are able to metastasize. So once again, we were very fortunate to get this grant from Swim Across America, which has been very, very successful in the research that Dr. Pratap has led. Again, my clinical practice mostly is for sarcoma patients, which is another rare type of cancer that is potentially something we will try to have Swim Across America help with some more grants in the future. These rare types of cancers are very hard to get funded studies up and running, and organizations like Swim Across America are really crucial to figuring out more about these rare diseases. I also have to mention how Swim Across America has impacted me personally, because I swim in the the event every summer. So personally, I am impacted because I am sore the day after every August when I do the swim. But it's a great event, and I highly recommend everyone listening to join, to spread the word to your friends and colleagues. The event every year is August 6th on the Ohio State Street Beach, and it's really terrific. I enjoy it very much, and I think everyone else would as well.
Thank you so much for that, Dr. Blank. And for our audience, Dr. Blank brings his entire family, and his kids and his wife are sitting on the beach cheering for him every step of the way. So every stroke of the way, I should say. Thank you very much, Dr. Blank. We are going to pass it over to Dr. Mackey.
Hi, I'm Carl Mackey. I'm a professor in the Department of Anatomy and Cell Biology. My lab studies breast and lung cancer, and we're trying to identify novel therapeutic targets in these two cancers, and the potential to target those for cancer therapy, as well as trying to identify markers of sensitivity in those cancers. So, for example, can you have a biomarker that can predict whether a cancer will or will not respond to these different treatments? It's another area that we're working on. And I've been blessed with a few Swim Across America grants, and it's helped me to continue with some of that work to develop some of the preliminary data to apply for larger grants, on which I've been successful based on some of the Swim Across America funding. So I'm very grateful for that, as well as publish some papers and make progress in these areas. So I'm Swim Across America has really impacted my research in a big way, and I'm just so thankful for it.
Well, thank you. We're thankful for everybody in this room and all of your teams, because none of this work can get done without you. So we're just so grateful to play a part in it. Thanks for sharing, Dr. Mackey. We will pass over the mic to Dr. Marzo.
Hi, I'm Amanda Marzo, and I'm an actual immunologist in the Department of Internal Medicine, and I'm in the Division of Hematology and Oncology. So my lab focuses on cancers, and in particular, metastatic breast cancer. So we want to know, so there's breast cancer patients come in, they go under various chemotherapy, radiation, immunotherapies, and some of these patients, like 20% of them will respond to treatment, and some do respond, but their cancers come back. And we're interested in those patients that maybe respond for a little bit, and then their tumors come back and metastasize. And we're particularly interested in those patients that don't respond to any treatments. So as an immunologist, immunology is very complex. So what we do is we study cells in the body that are basically there to eliminate any infections, any cancer. So if you think about it like an army, you've got your troops that go in and then you protect your country. So we've got cells in our body that protect us from things from outside and things from inside that can go wrong. And with any army that goes into battle, some of these enemies cross the line, and we have cells that can, if you think about it, in our body that can keep them at bay, right? Keep the cancer cells from metastasizing, or keep them from even forming. So we study two types of killer cells, right? Those that arrive at the scene first, and they're what we call natural killer cells. And they try and keep everything at bay, but often they need backup. So our backup cells come in, and they're our cytotoxic T cells, and they try and get the job done. But with metastatic breast cancer and those that don't respond, those cells can't even get into the tumor. So we're interested in studying how to get them in. And then once they're in, make sure they can function, make sure they're armed and they can do their job. Because a lot of the times they become disarmed, dysfunctional, and then the tumor metastases, and it goes to the lung and the bone, as you heard from Dr. Blank. So I've been blessed by getting a few Swim Across America grants that have, in the years, enabled us to keep working on this. And I'm really happy to say that we've put in some grants for national funding using preliminary data that we obtained from these grants from Swim Across America. We'll hear by the end of the year, early next year, whether these three will get funded. But more exciting is that we've actually developed a therapy in a mouse model to start with that we can establish metastatic breast cancer and we can eliminate it. And we've got 2 papers on the burner that we should have out by September, showing that, those patients or those tumors in mice that can respond but then come back, we can cure them, and we can also cure those that actually don't respond to therapy, and we can get those cells in and do their job. So the next step for us is to get more funding and to actually do this in patients or do this in what we call a humanized mouse model. And Dr. Pratt will actually cover a little bit on this. And basically, it's a mouse that we give them a human system, and then we can use human cancers, put them in and follow these things and test our therapies out on them before we actually go to clinical trial.
That is incredible, Dr. Marzo. Thank you so much for being here today and for sharing that. It sounds like it's a long road, right?
It is a long road, but there's light at the end of the tunnel for us. And we thank Swim Across America and everyone that donates to be able to actually get us this far. And it should be exciting stuff going forward, especially in our lab.
That's just fantastic. Thank you so much. And last but certainly not least, Dr. Pratap.
Thank you so much. Hello, everyone. I'm Jitesh Pratap, an associate professor in anatomy and cell biology at Rush. And my sincere thanks to Swim Across America team. And, fortunate enough to get some Swim Across America grants in collaboration with Dr. Blank. And so our focus is to understand when cancer cells, they move from the, one tissue to the other tissue, such as bone, how they change, what extra changes they acquire, so they survive in the other tissues such as bone metastases. As Dr. Blank mentioned, that right now there are very limited treatment options for those patients. So what we are trying to do to understand going into the molecular pathways, what are those proteins, what are those genes which are going up or down, and looking into these, can we target these cells when they arrive in the bone microenvironment? So with Swim Across America funding, what we see in the breast cancer bone metastatic cells, when they change one location from one to other location, they induce their recycling process. By recycling their components, they survive for a longer period of time. And while doing that, they change their cytoskeleton. So any support system for this recycling process in the cancer cells really induces. So that's the very exciting finding we found, and we are now with Dr. Blank trying to use more patient samples to take it to the clinical perspective in this finding. And with Swim Across America grant, we have recently published 2 research articles with Dr. Blank and then applying federal funds. One we already have a small grant from and in coming months, we're gonna be putting these data together for the larger federal funds. And once again, thank you everyone for joining and supporting our research.
Thank you so much. And I love that you brought up that you and Dr. Blank work together and collaborate, right? You're in 2 different types of medicine, but cancer brings you together. And that's just really, it's a beautiful function of collaboration and really working with your colleagues for the greater good. So fantastic that you're both with us here today to talk about that. Thank you, doctors, for the very informative and impactful introductions. We're going to shift gears a little bit. We're going to talk about why you're passionate about cancer research. Right? You are doctors, you treat patients, you do clinical trials, you do that research, and you want to do that to further cancer research. But what keeps you coming back every day? So Dr. Bishehsari, please come off of mute and share more about why you got into cancer research. And like I said, what keeps you coming back every day? Because it's not it's not a short road, it's persistence and it's perseverance. What is it for you?
You're absolutely right. Actually, for me, I think it's not a difficult question to answer. As a GI gastroenterologist, if you want to make a change, and you're interested in cancer in general as a leading cause of mortality, the two cancers I see is colon cancer, which is the top 3 most common cancers in the US, and pancreatic cancer, which is the deadliest cancer. And so in order to make a change beyond the patients that you see on a daily basis, is to keep this challenge on and go after them. So that's the big motivation that I have for studying these cancers.
Wonderful. Thank you. And thank you for everything that you do. Dr. Mackey, how did you get involved in molecular level cancer research? And what is your motivation to continue doing this work? And what brings you back every day?
Well, I think the motivation for me, there's really 2 things, I guess, one more important than the other. The first is that, we're scientists. So we get to come in every day and decide what research area we're going to work on. No one's going to tell us what we have to work on. We get to design experiments and carry them out, answer a question. So it's really intellectually stimulating in that way. And then when you see those cancer cells on the plate, dying, or you see that tumor in the mouse begin to shrink from the treatments you've been doing, the experiments you've been doing, that's really rewarding. So that's the first motivation, I guess. But the second motivation, more important, is wanting to have an impact. I think the statistics are something like 1 in 2 men and 1 in 3 women in their lifetimes will get cancer. I mean, it's a staggering statistic, right? There's so many people. And in my family, my father died of lung cancer. My cousin had lung cancer. My grandma died of colon cancer, and I don't know who's next. And, just so much suffering, though, and you can see the impact it has on families. Recently, we found a family friend. She's younger than I am, but she has 2 young daughters, 8 and 10 years old, and she found out she has breast cancer. So, she started treatment. So there's a lot of suffering, and we want to have an impact. We want to try to help people and make our work impactful and important. And I think that's the second motivation.
Absolutely. And what I'm hearing is a tagline for Swim Across America, right? You provide families with hope every day that you come in, and you're finding that tumor shrinking, and Dr. Barua with his early detection, like, we are providing more families, more people that we love with hope. So keep doing this amazing work. We just can't thank you enough. And thank you for sharing your personal story with us. Dr. Barua, if you can come off of mute and share what your passion about early detection testing in ovarian cancer is, and what got you into that work, and what keeps you coming back every day.
Thank you very much. This is the most valid question that I always encounter. For me, it was kind of serendipitous finding. I was working on ovarian function, and then at some point I found that some of my animals with ovarian problem developed ovarian cancer. And immediately I searched in the literature in the human, and I found that there is no early detection test. And because this is a very fatal malignancy, most of the women dies because there is no detection test. And then we found that normal procedure now, we call it CA-125 in the blood, and traditional ultrasound are the common treatment or the detection modality. Unfortunately, the serum level or blood level of CA-125 is not specific for this cancer. It can go high for various benign reasons like endometriosis, like fibroids. And on the other hand, the ultrasound currently available cannot detect the changes, early changes associated with the ovarian cancer. So unfortunately, none of the researches were possible to detect because there is no preclinical spontaneous animal model. As you know that in many researches, we use rodent like mice, rat for our research. Unfortunately, these rodent models, they do not develop ovarian cancer spontaneously. So when you induce a tumor in these animals and the data you get, it is very difficult to translate those data into the clinic. So that is one of the reasons that we are trying to generate this data from an animal model that is spontaneous for this ovarian cancer, and this is very showing remarkable similarity to our human patients. The third thing is, I was so surprised that if we can detect the disease early, I can save the life of patient. Let me tell you one of the story. I have a colleague at Rush who is she was a gynecologist, OBGYN, and one day she was telling me that she was diagnosed by a CAT scan. I said, what? Because CAT scan, there's no CAT scan. So she was saying that I do ultrasound my patient every day, but unfortunately, she even herself, being a physician, couldn't diagnose herself that she is developing a tumor in her ovary. So how she detected? When she was sitting on her couch, her cat jumped on her belly, and then she feel pain. So then next day she came to the clinic and she scanned and she found a tumor in the ovary. So this is what this does. So if we can detect, we told that as an what it said, that happenstance detection. So meaning if we can detect the tumor in the ovary, 90% patient can survive. So that is one of the reasons. As you have seen, most of the disease, most of the drugs available for ovarian cancer are, I mean, the patient develop resistance to these drugs. So the main problem of high rate of death due to ovarian cancer, it come back frequently, it recurs. So meaning none of the drug available has any efficiency to prevent this this cancer and save the life. So most pragmatic way to prevent the incidence or to catch the disease at early stage, and that's the reason we need an early stage early detection test. And it's very easy because I believe, and we see most of the women goes to clinic once in a year. So if you just take a 1 ml of blood from the woman, and then if you send it for detection, the blood marker level that we have, and we can easily monitor this patient. Okay, let's monitor you through the ultrasound that we are developing. And we have developed an imaging agent through this swim across funding. When you inject this, and immediately it will increase the resolution of the ultrasound so that it can easily differentiate normal ovary from the tumor ovary. And you can catch the disease when it is remained in the ovary. By that way, we believe that the specificity and sensitivity, if we can improve, it will help us to detect the disease at early stage. You may know that when a woman has a pain in her belly, she goes different doctors. Like she may think, oh, I have maybe cycle problem or I may have stomach problem. At 6 months, when visiting most of the clinic, there is no disease detection. Then she comes to GYN, gynecological malignancy or oncology clinic. Then by this time, her cancer is spread at hole in the belly. So you cannot save this patient. Whereas if you would have some treatment or detection modality, then you can detect her, you would have saved her life. So that is one of my most passion, that if we can detect, if we can establish something, we can save the life of a woman. And I believe it will be possible in nearest future. The panel that we have got, we just need one more clinical study so that we can comment on the sensitivity and specificity of the marker that we develop, as well as our ultrasound contrast imaging. I believe that I made it clear why it is my passion. Thank you.
Thank you so much. And you're absolutely right. Your passion is so clear. And as a woman, I'm so grateful for the work that you are doing in this area. Thank you so much. Doctors, thank you for sharing those answers. I have one more doctor I want to hear from on this question. Dr. Blank, same question to you. You know, why do you keep coming back every day? Cancer work and cancer research, it's not easy. You might fail sometimes. You might have to redo a test, redo a project, redo a study. But what keeps you coming back?
Well, you're right. It's hard. It's a lot of ups and downs. You know, my passion for research really comes from my clinical practice. My clinical practice is 95% of my life here at Rush. I have a very busy clinical practice, seeing patients, doing surgeries, And I see in my practice that things aren't perfect in the clinical treatment of cancer. It's just not. We have patients who have tumors come back. We have patients the tumors grow on treatment. We have patients the tumors spread places. And we do the best we can from the clinical side, but we just don't have all the answers. And it's very humbling to see sometimes, even when we give it our best, bad outcomes can happen, and that leads us to ask questions for, well, why is this happening, even though we did everything by the book, by the NCCN guidelines, and it leads to questions, and those questions lead to working with brilliant minds such as all the other people you see on the screen here who can come up with questions at a basic science level and ask the question and formulate a hypothesis and come up with a plan of attack. So it's important. Like I said, 95% of what I do is clinical work, but because I see where we could use improvements, I really squeeze out that extra 5% into a research capacity and try to come up with better answers for the patients that I see in the office. And again, thanks to grants like Swim Across America, a lot of these are possible. Like I said before, the cancer I deal with mostly is sarcoma, which is a rare cancer. There's only a few thousand every year in the United States. And without grants such as the Swim Across America grant, it's very hard to get funding for projects investigating these things. And I think the future of figuring out how to better the treatment of these patients starts with kind of seed grants such as the SWIM grants. But not a day goes by in my practice seeing patients or going to tumor board that I don't think of some other sort of research question that pops up that comes from, hey, this didn't go as well as we'd like. Why didn't it go as well as we'd like? So in short, I guess the clinical side of things and seeing the ups and downs of the clinical side of medicine is what fuels my passion to try to make things better.
Sounds like the good outweighs the bad, and the challenge of there isn't one patient that's exactly the same as the next. And so I'm assuming it keeps you on your toes and gets that collaboration going, because as patients and non-doctors, we look at doctors like you're supposed to have all the answers, right? And you're all humans just like all of us, and that's not always the case. So next time, audience, when you go and see your doctor, give them some grace, give them some pause. They're doing the best that they can to find the answers that you need. And just know that there's so much work and passion that goes behind everything that these doctors do. So thank you. Thank you for sharing that with all of us. Dr. Marzo, Dr. Pratap, and Dr. Mackey, in that order, could you please share what you think are the most exciting recent breakthroughs or breakthroughs that are on the horizon of what we're going to see in cancer treatments? So Dr. Marzo, if you wouldn't mind going first. Oh, you might want to just take yourself off mute.
Okay, it's all right. So the first one that comes to mind is we now have a platform that we can use because of the COVID vaccine. So when COVID hit, the government poured a lot of money, industry poured a lot of money, and scientists around the world worked together to produce this vaccine in a record amount of time. Normally that would take 5 to 10 years. We get it in a year. So now they've developed this platform that we can now use for other diseases, and one of them is cancer. So there's a lot of companies out there, a lot of researchers that are taking this technology and applying it to cancer. And so I think the most exciting thing is in a year or so, we'll start to see some of those therapies coming out using that technology.
That's amazing. And, in a recent conversation that Dr. Marzo and I had, she shared, like, look at what scientists and doctors were able to do in such a short period of time with the COVID vaccine. And that took approvals, money. How important is that, shaping what we can do for other treatments in cancer if we had that same kind of escalated timeline?
I think that's the problem, is scientists are restricted. We get most grants, it's 10 to 15% of grants get funded. So there's a real big cutback. But with COVID, they poured so much money in that you could see what scientists are capable of doing if you give them the money and you give them the manpower and you're able to let them collaborate with industry, scientists around the world on the same topic that came together, and we got the COVID vaccine. So you can imagine if they did the same thing with diseases like cancer, arthritis, and things like all those autoimmunity, if they funneled a huge amount of money into a group of scientists to work on that, we would be way further ahead, I think. And money is the bottom line. So Swim Across America helps us do that, get one step further.
Fantastic. Well, thank you for sharing your insight. Dr. Pratap, curious to know, in a recent conversation that we had, we talked about the Pdx model and what that means. So if you wouldn't mind sharing a little bit more about that with our audience.
Sure, absolutely. I think this is the one approach called PDX, which is patient-derived xenografts. And I'm very hopeful with this approach is going to change or revolutionize the cancer field in many ways. So in short, what you do, you take a small piece of tumor from patients and put it in a mouse. So what happens in mouse, these tumor cells, they grow very rapidly. So what you're doing, basically looking the disease within few months rather than years. So in doing that, we take the small portion of these tumor back from these animals and then look at the molecular level. And that's how we can see how tumor is changing or evolving. So that's a big insight that we can have, that how patients are gonna respond and how tumors are gonna develop in patients, so that we can really tailor our treatment plans. The one biggest advantage is that in these animals, having these patients' tumor, we can test various drugs based on how these cancers are evolving, right? So what you're really doing, they narrow down the treatment plan based on the changes like mutations happening in these tumor tissues. So that's very, very exciting. In that way, you are more precise in the treatment plans. You can reduce the unnecessary chemotherapeutics, which is not going to work in patients. So at Rush, with Dr. Blank's help, we are trying to develop the similar thing for the bone metastases of the breast cancer. And what we are basically looking into that, when breast cancer go to bone, it changes the recycling process, as I was earlier mentioning that. And for this recycling process within the cell, it really changes the skeletal component of the cells. So what we are looking into that, can we target this skeletal component and then reduce the side effects of the chemo and increasing the effectiveness of the chemotherapeutics. And not only that, also identifying the new marker proteins, which can really kill the cancer cells when they go from one place to the other. So this Pdx model, I think our panelists here, they are using for their various different types of cancers and testing their relevant targets. So I'm very hopeful with this approach, and with Swim Across America, we are trying to develop it here too. Thank you.
That's fantastic. And, just to give our audience some concept of time, what were you seeing 5 years ago or even 10 years ago in this field?
Yeah, so I think that the PDX is basically started to develop in various institutions, and some companies are also trying to develop, but there's always a limitation with getting the patient sample, right? The fresh patient sample, which is the critical most point here to develop these PDX animal models. So now I think few years in this approach, my guess is another 2-3 years, probably we're going to develop a very good data set that what drugs are going to work for which type of cancer, or which are more effective for treating the specific changes in the cancer mutation. So that's how I think we are going to be more precise in treating the cancer. So my hope is like 3, 4, 5 years, very soon.
That's fantastic. And it goes to what Dr. Marzo was talking about earlier. There's the light at the end of the tunnel. This has been years and years in the making, and now we're finally seeing that light at the end of the tunnel. So thank you so much for sharing that with us. Last but not least on this topic, Dr. Mackey, if you couldn't mind, tell us more about the public databases and the cross collaboration that not only happens within Rush, but across networks of scientists.
Yeah, I think that what we're talking about is the, what I feel is one of the biggest breakthroughs that's come along, that's sped up research, and that's the availability of information. There's so much information now that's freely available to cancer researchers, in all these different databases that are being developed at different big research institutes like at Harvard and MIT and MGH and these different places. What they have, these different databases that are available. Things, and here's just to name a few. One is the DepMap database, where people have knocked out every single gene in almost every cell line available, and measured how important it did knock out of that gene kill the cell, or was the cell able to survive this or that cell. That's just one example. Another one is called the Genomics of Drug Sensitivity in Cancer database, where they've taken every single, I don't want to say every single, but let's just say hundreds of cancer cell lines or different cancer types, treated them with every drug imaginable, and measured the response of those cancer cells to those drugs. And then there's another database called the Cancer Cell Line Encyclopedia, where they've taken, these different research institutes have taken every single cancer cell line and basically determined all the gene expression profiles in those cancer cell lines. So how does that speed up progress in cancer research? So here's an example here from my lab. So our lab is interested in, currently, we're trying to identify the responses in lung cancer to a specific anti-cancer drug. And we want to know, for example, if these lung cancers don't respond to the drug, then what genes are associated, what gene expression patterns are associated with poor response? Or if these lung cancers do respond, can we identify gene expression patterns that are associated with good response? So in the old days, not too long ago, what we would have to do is write a grant, and we would write the grant and say, okay, so what we're gonna do in this study is we're gonna gather up all these cancer cell lines. We're gonna treat them with this drug. We're gonna measure their response, and we're gonna categorize them responsive or non-responsive. Then we're gonna do an RNA expression profile in these cells, and we're gonna try to identify the gene expression profiles between the responsive and non-responsive cancers. And the whole process is gonna take about 3 years. Right? We don't have to do that anymore. Now what we do is we go to this Genomics and Drug Sensitivity in Cancer database, and we find cell lines that are responsive to this type of drug or are non-responsive. It's all there for us. Then we go into the Cancer Cell Line Encyclopedia, and we get the gene expression patterns in these cells. And now we can compare those gene expression patterns. And then we can identify potential genes that are associated with good response and poor response. And that whole process takes about a couple days, because all that information is available, because you have to have someone good in your lab like I do, who's able to carry out all those analyses. But now instead, that process is just so sped up. Now we can go after try to validate these gene expression profiles and their association with good and poor response. And we basically cut out 2 to 3 years of work. So
that's incredible. That cross collaboration and the sharing of knowledge, right? What I'm hearing is that, the enhanced collaboration and sharing that knowledge across medical institutions and across scientists will continue to make those breakthroughs and cures more frequent and more of our reality, rather than having to wait that 2 or 3 years. So it's really heartwarming to hear. And it's, again, that sign of hope. We are moving in the right direction. Really appreciate that knowledge that you just shared with us. We're going to move on to a little bit more of a concerning question. unfortunately, there are these barriers of entry into cancer research, one being funding. We talked about that a little bit, compensation for the scientists, timing and approvals. And so, Dr. Bishehsari, if you had the opportunity to make or change a policy or a recommendation that would speed up the process toward cancer breakthroughs, what would that look like, and what would that be?
Sure, I'm happy to address that because I think that's the same concern of the old panelists sitting here. I think we need a big change at the policy-making levels. Government funding for research has plateaued for a couple of decades, while the prices of everything, as well as the number of science graduates, went up exponentially in the same time. 20% of the NIH funding goes to 2% of investigators in certain institutions. So as a result, if you put yourself in the shoes of new investigators coming about, they are very hard to find because they are concerned of their future competing with so many others for a little money to use their creativities to fund their creativities. So new grads tend to go to industry, pharma, some sort of private sector, and very small fraction may consider staying in academics as a path to become a PI or a scientist. And they know, if the ones that stay, they know the first independent funding from National Institute of Health or other governmental funding agencies would not come until they are in mid-40s, while the peak of enthusiasm and energy to make changes occur in the 20s and 30s. So research, especially in diseases that don't affect everyone, like COVID. COVID affected everyone. So that is okay to leave it to industry, because industry thinks, or private sectors think of margin in short or mid-term, but developing care for these rare cancers like sarcoma, as Dr. Blank was mentioning, or pancreatic cancer, which affects 10 to 15 people in 100,000 population every year, may not give a huge benefit margin for the private industry. So it is, I think, the responsibility of the funding agencies to increase the dollars and distribute it efficiently and in a timely manner, using the infrastructure that we already have in place across the country, and training new investigators and funding them sooner while they are still enthusiastic.
Fantastic. Thank you so much. And, on that note, Dr. Mackey, where have you seen, the passion for young investigators and this next generation of doctors? Do you see that they're passionate like everybody on this panel, or do they need a little bit more to get them as passionate? What are you seeing with your students?
Well, I think there's a lack of passion, kind of what Faraz, I think, was getting at. The students don't necessarily, they see the struggles that we have every day just trying to secure funding. And, they don't necessarily want to choose that path. As intellectually stimulating and potentially impactful as it may be, it's going to be a very difficult life to do that. So I just had a student graduate, and she went into industry. It's you don't have to fight for grants there. So I think that what Faraz was saying is absolutely true. We need to build up more motivation for these young scientists so that we can keep allow them to build up their creativities and keep that pool of scientists, keep a pool of scientists alive to continue this work.
Well, they've got great mentors and teachers, so it shouldn't be, in theory, it shouldn't be that hard. But I hear you, that driving that passion and really getting the next generation to care just as deeply as you all do is an ongoing effort. So anybody in the audience that's looking to get into this work, please consider it, because it truly is very impactful, and the passion is never ending. So I have been monitoring the questions that have come in through the chat, and I've incorporated them into some of our discussion so far. But one that we haven't quite covered yet, that has come in a bunch of different ways is kind of our daily habits, right? What can prevent us from getting cancer? What can we do, you know, aside from early detection, aside from treatments, our regular everyday? How can we be better as humans, and so that we don't have to have a cancer conversation about ourselves or a loved one? So I'm going to ask Dr. Barua to start us off in that discussion.
Well, thank you very much. It's a very, very, very important question, because you know that literature shows us that 60% of cancer can be prevented by a good behavior and lifestyle. And when I mean this behavior and lifestyle, it includes eating habit, food, sleep, taking rest, enough vacation time, as well as how your social life is. So with all those habits, definitely, as one of the example is smoking. So all of us know that smoking helps to have the lung cancer, not only lung cancer, some other related cancer, as well as other non-malignant diseases. This is one of the examples. Suppose obese. Obese is a factor that is associated with most type of cancers. All of my colleagues sitting here, they know it very well. So preventing food habits that make a person obese, so it also helps preventing the developing of cancer. And similarly, you know that we also know about the balance between, we call it the day and light cycle. So the hour you sleep, the hour you work, it also affect in the long run. As you know, cancer is not a disease of developing tomorrow. It takes time. Over the period of time, at in our body tissue cell level, the changes happens with response to behavior, food habit, sleeping, or health and hygiene. This is very important. Even the environment in which we are, so this very important. So that's the reason they say that 60% of cancer can be prevented by good behavior and health and hygiene. Why I'm saying that? Because awareness, meaning all these things come to a point that all member of the society, or all of us, need to be aware, or need to make someone aware what can cause cancer. It may be possible to do some awareness work in the community, in the school, or whatever um, social activity that we do. Awareness is very important, so that it can be, there are many cancers that can be prevented, like the one that cervical cancer coming from a microorganism, or there are some other cancers coming from infection. So these are the ways that you can prevent by awareing all members or everyone to have a good habit, health and hygiene, to have a good food habit, sleeping, and maintaining their proper day and night cycle. I believe this will contribute a lot in prevention of our disease. As we know that if when someone, although there may have some responsive drugs, but please, I like to remind you that drug is itself toxic. All cancer drugs are toxic. It not only kills the tumor cell, it also affects the normal cells, meaning it reduces quality of life. So it not only costly, it also affects the quality of life. So prevention is better than cure. That's what I will go for the awareness. Thank you.
I love that. Prevention is better than a cure. We're working toward that cure, but we can all do things in our daily lives to make that prevention easier for us. I'm going to ask Dr. Mackey to summarize, what you tell your students, and because I love what you said the other day when we spoke. So I'm going to pass over the mic so I don't steal your thunder.
Okay, yeah. We have this discussion at the end of my cancer biology class every year. It's like, what can we do, or can lifestyle choices affect cancer incidence? You bet, it absolutely can. So I think there's three things that I tell the class, and the first one is the one Dr. Barua just mentioned, was smoking. Obviously, we know smoking is linked to lung cancer, no question about it, and so many other cancers. So we say to the students, don't smoke. That's number one. Then there's anecdotal evidence out there linking stress levels to cancer. And so I show this data in class where researchers took a mouse. They took a tumor and they grew it in two different mice. And then they let one of the mice just run around the cage happily, right? And the other mouse, they put in this tight little space. And it was sitting there, the mouse was like all stressed out. It was called constraint stress. So it just put him in this tight space where it couldn't move, and and stress hormone levels increased in the mouse and all this. And then they looked at the growth of the tumors. So in the mouse where the tumor was, that was just able to walk around the cage freely, the tumor grew at a certain rate. But in the mouse that was stressed, the tumor grew much quicker, grew to much higher levels. Then they treated those mice with an anti-cancer agent, and the tumor in the mouse tumor in the stressed mouse did not respond to the treatment. So I tell my students, don't stress, be happy. It's another way to avoid, cancer. And then I show this data about obesity, linking obesity to cancer. There's links, strong links with obesity and cancer risk because of, high levels of insulin and insulin-like growth factors circulating in these patients, that have potential to drive cancer growth. And so I say to the students, be healthy. So at the end, I tell them there's 3 things: don't smoke, be happy, be healthy. That's how you can avoid cancer.
I love that. Not all easy. It's easier said than done, but if we can all focus on that and be mindful of what that means for our health and long-term health, it truly is a mindfulness practice, right? There's stress every day. And how do we reduce that? You eat healthy, you're going to feel better. You sleep better, you're going to feel better. You remove some of those toxins that don't need to be in your body, you're going to feel better. So I love that. Thank you so much for sharing that. It's so motivating and inspiring to me and hopefully all of our audience today. We've got a few minutes left, and I just wanted to end with, and anybody can answer this question, what do you see as the future of cancer care? When I said anybody, I'm going to point to Dr. Blank to start us off, and then we'll have one more doctor. Dr. Pratap, if you wouldn't mind sharing your thoughts as well, and then we'll have a few closing remarks, and we will close out our wonderful discussion today.
So in short, I think the future is at the molecular level. I have a mentor who once said, we've gone as far as we can with removing cancers with an actual scalpel, and at this point, it's up to the molecular level scalpel. So targeted therapies, more and better research like my colleagues here are all doing, and finding very specific medications, targeted medications to attack these cancer cells, and attack them alone rather than attack the rest of the body as well. And once again, there's nobody better at helping to figure this out than everyone you see on the panel here.
Fantastic. Thank you, Dr. Blank.
I totally agree with Dr. Blank and some of our panelists, as they mentioned that as last few years, the cancer researchers, they started to build that database, which basically help us to ask the right question and understand the how these cancer cells they change and evolve, and how we can find these potential targets to kill cancer cells. So I think in a few years, the precision medicine probably is the future, where we can really not only target cancer cells, but also the microenvironment, the cells which support these cancer cells like immune cells, and how we can make it more active or more responsive to that. So I'm very hopeful.
I love it. Hopeful, and everybody's putting in the work on this call to make sure that we have prevention, early detection, cures, and keeping our patients and our community happy, healthy, and not smoking. Audience, I want to thank you for being with us today and for such great questions. Thank you to everyone at Rush, all of these incredible doctors, also Aaron Schlepp and Laura Raymond and their entire teams. And of course, everybody you see on the screen. We can't do this work without you. Who are fighting, you all are fighting cancer in a way every single day and showing up for your community and all of us for a brighter future in cancer care. We are so grateful for that, and you truly are an inspiration to all of us. We hope everyone found this discussion inspiring, and if you are able to, we would love for you to join us in our mission in the fight against cancer by joining our cause and fundraising with us as a volunteer, a swimmer, a golfer, or just as a cancer advocate. You can join us in any way that feels good to you. And you can do that by visiting our website at swimacrossamerica.org/Chicago. And just know that all donations and all of your support go directly to Rush University Cancer Center and directly impact the doctors that you see on your screen today and their teams. So one more time, that is swimacrossamerica.org/Chicago. It's been a pleasure to be with all of you today, and I look forward to seeing you at the Ohio Street Beach on August 6th, where you will find Dr. Blank and hopefully some of the other doctors on your screen. We thank you very much, and we hope you have a wonderful day.
Thank you very much.